I'll introduce you guys briefly.
Okay, great.
Hello, everybody. We're very glad you could join us. If you were at the webinar last week, welcome back. If this is new for you, we're delighted you could be here. My name is Michelle Mannion, and I'm here from the PCD Foundation with Lynn Ernie. So we will be the tech people. Actually, Lynn will be the tech person. Don't send things to me. I won't be able to help you. So if you have any issues or logistical issues, please let Lynn know. As we move into the program part of this, if you have questions, please put them in the questions and answers section, and we will get to those at the end of the presentations. Just as a little introduction, I am the executive director of the PCD Foundation, and as many of you know, the PCD Foundation is currently the only North American patient advocacy group focused on PCD. And in that role, when we started 22 years ago, our goal was to get to clinical trials. Our first two programs were called the Path to Clinical Trials. So we are very much in support of clinical research, understanding that that is the only way we will make improvements in quality of life and expand life expectancy and make life better for patients with PCD. So we are completely supportive of trials. That said, when we provide information like this, it is only provided as information. It should not be seen as an endorsement. When you are interested in participating in a trial, this is something you should be talking about with your own personal physician to see if it makes sense for you. So with that, I'm going to introduce our speaker today and a panelist that we have from ReCode. First, Dr. Ramona Doyle. She was with us last week. Dr. Doyle is an experienced academic clinician scientist, educator, and biotech executive who understands the life cycle of drug development. She has therapeutic and development experience with small molecules, biologics, and cell-based therapies for unmet medical needs. She is passionate about science and about patients, and she understands what is required for innovative therapeutic product development in diverse patient communities. And in this case, we're talking about a rare, very rare patient disorder. Dr. Doyle will be the presenter today. And on the panel, we have Dr. John Matthews from ReCode. He is the Senior Vice President of Clinical Development. He brings over 20 years of proven leadership in clinical trial design and drug development and extensive patient care experience. Prior to joining ReCode, he was at 23andMe, where he was the Senior Clinical Development Lead for Rare Disease Therapeutic Strategy. He has a very impressive resume from prior to 23andMe, and I'll let you guys look that up on your own on ReCode, but we definitely know this is someone who's been involved in drug development for rare disorders. Dr. Matthews trained in pulmonary medicine at Royal Brompton in London, and the Lane Fox Respiratory Unit in St. Thomas's Hospital, London. He also has a PhD in respiratory clinical pharmacology. So with that, I think we will turn it over to Dr. Doyle.
Good morning, and thanks for that kind introduction. Lynn, I guess we're going to talk slides if you'll let me know when you get the first slide up. So I'm a clinical professor at UCSF. I'm also a trained pulmonologist, and I'm really, really excited to be able to talk with you today about what I believe is the first trial for actually correction of the underlying problem in PCD. So I'm also a paid consultant for ReCode Therapeutics. Next slide. That's my disclosure slide. This isn't, as already stated, this is an educational session. We're going to really focus today on the phase 1 trial of an investigational product. And you've already heard the disclosure that the views expressed in the webinar don't necessarily express or reflect those of the PCD Foundation. But I know the PCD Foundation has been fantastic about trying to get new therapies to patients. Next slide. So in the discussion today, and we're going to have a lot of time for Q&A, I'm basically going to provide a little introduction to ReCode Therapeutics, which is the sponsor or the company that's performing the trial and has developed the therapy. I'm going to talk a little bit about the mRNA therapy itself, which is also called RCT 1100. I'm going to give a basic overview about the trial, who can participate, and what to expect, and then be sure that you all have contact information about how you learn more about the trial. I will say that detailed information about the trial, if you have any interest and are potentially eligible, ReCode I know has people that are anxious to provide even more information than I probably will have time to do today. But this is meant really put down your pens and pencils and just a broad overview. There will be a lot of information available to you from other sources. So first slide, next slide. Lynn, do we have the next slide? There we go. So, excuse me, I think we all have maybe a few little coughs and colds this time of year. ReCode is a company that's devoted to rare lung diseases, and in particular, those that don't have any way to actually treat the underlying disease. The lead program right now, excitingly for you guys, is in primary ciliary dyskinesia. And the vision of the company is they want to make a transformative difference for patients with genetic diseases, including and focused first on PCD. Next slide. So the question would be, why primary ciliary dyskinesia? Well, first of all, is there a need for new therapies? And definitely, this community is an underserved patient community. As many of you know, or maybe have experienced, many patients go undiagnosed, and many people who are diagnosed are often told it's not serious. The progressive nature of the disease and the main impact being damage to the lungs can lead to serious impact on quality of life and further lung damage. And it sets up this vicious cycle of damage to the lungs, which generally only progresses. Now, there are symptomatic treatments to deal with the problems of excess mucus production, et cetera, but they don't actually address the underlying cause. And those treatments, by the way, are very important. And, you know, that they have been successful in helping patients a lot with PCD. But there aren't really any cures for PCD or targeted treatments that get at the root cause. And management, as I mentioned, is focused on the symptoms, recurrent infections treated with IV antibiotics, bronchodilators to help clear sputum, pulmonary clearance maneuvers, etc. So that's why PCD is an important disease. So let's talk about the developing mRNA treatments. And M just stands for messenger. And if you can remember that that's what this product is, that's I think a good way to think about it. So the cells make messenger RNA, all of us have messenger RNA that actually provides instructions to cells to make proteins. And that's a common thing in all the cells in our body. In an individual with PCD, a genetic mutation or a change in the underlying genetics leads to the mRNA being insufficient. In other words, it provides a faulty set of instructions, and that leads to a protein, one of the many proteins that's responsible for the structure and function of cilia, which are the fine hairs that are very important in many functions in the body. The cilia are either missing certain structural supports, or they don't work properly. Across all of PCD, there's actually a lot of proteins that are missing or defective. And what the result in that is that the cilia don't beat properly, they don't move properly, and they don't perform their function, which is in the respiratory tract, is to move mucus along and keep the lungs clear of infection. We know currently, and this is a very active area, that mutations in nearly 50 genes are known to cause PCD. So exciting, thanks to a lot of hard work over many, many years, we can now make messenger RNA in the lab. And at ReCode, messenger RNA is made that carries the correct set of instructions to the cells to make the specific proteins, including those that are related to PCD. And this is a technology, by the way, the use of messenger RNA that has been used successfully in other diseases. The message is actually delivered in an envelope known as a layer of lipids, known as a lipid nanoparticle. And so based with the mRNA constructed in the lab, put in the envelope of the lipid nanoparticle, it can be delivered excitingly directly to the lungs in this case. One thing to know is that all we're doing is tapping the cell on the shoulder and saying, make the right protein with the messenger RNA. We're not going in and altering the DNA, the the Uber section, basically the large genetic informant of all of these processes. This is simply along the line passing the message to the cell, you need to make this protein. So given that this is not a treatment that permanently affects the DNA in your genetic makeup, you will need to be dosed regularly to keep telling the cell, make the new protein. Now, the mRNA treatment and the instructions that go with it have to be specific for what that particular issue is. As we mentioned, there are many different mutations in genes that can cause PCD. So next slide. Fortunately, the technology has advanced so that you can make the instructions specific for the protein, for the problem and the genetic problem that leads to it. So in PCD, about 10% of people diagnosed with it are missing a protein called DNAI1. In people without PCD, DNAI1 protein is made from DNAI1 messenger, and things move along quite smoothly. But if you're missing the protein, you can be given, your cells can be given instructions to actually make the protein and restore the function of the cilia in the lungs, which then gets at the root cause of PCD. So just as a quick to look at some of the illustrations, where do cilia lie? They are on the tops. They're on cells that help, as you can see in the picture on the far left, move mucus through the airways. And if you take a cross section of one of those little hairs, you can see how complex the structure is. You could imagine to create a beating, moving entity. There's a lot of structural support involved. In the particular case of the DNAI1 mutation, it's the outer arms, which you can sort of see with the X's over them, or the outer dining arms, where there's a problem. And you can see that in the case of the dysfunctional or unhealthy arms, that the cells, the whole operation, the whole machinery doesn't move properly, and the cells don't move at all or don't move properly. So a healthy ciliary axoneme, which you can see, which is basically the cross-sectional view on the right, has functioning, and those are the arms in red, I believe, the healthy ones. So this, by going to the level of the instructions to the cell, you're improving the root cause of the dysfunction that leads to the fact that the cilia don't move properly and hence cause a lot of problems in the lungs. Next slide. So I'm gonna share somewhat briefly the design, a little bit about the trial. Dr. Matthews is online, I think, for specific questions, but also realize that if you're potentially eligible and interested, and you and your doctor discuss it, once you decide you would like to be screened for the study, a lot more information, a lot more detail will be made available to you. And certainly if you want to proceed with the study, they'll be sharing a lot more information with you. So don't feel like you have to remember all this now or have it all down. Next slide. So the phase 1 study to assess RCT 1100 in DNAI1 PCD patients is an open label study, which means everybody in the study will actually get a dose of the new therapy. And this is only a single dose, by the way. Other studies will follow around repeat doses, but it's only for a single dose via a nebulizer. And I'm sure many of you are familiar with nebulizers and inhaled therapies. The study visits are set up to monitor for safety, and your convenience and the amount of disruption to your lives is certainly something kept in mind, but you are, for the purposes of the study, asked to return after the first study. And the first and only nebulization. The study includes one overnight stay on the day that the drug is administered. That's so you can be watched closely and information can be obtained to be sure things are going well. So the goals of the study really are to see how safe and well tolerated this nebulized messenger RNA treatment is, to determine if any parts of this therapy, once they're inhaled in your lungs, are actually detectable in the blood after you have one nebulized dose. And importantly, also markers in the blood to assess any immune response to the therapy will also be followed over time. Next slide. So again, don't worry about writing all this down, but just basically in terms of inclusion and exclusion criteria, inclusion is needed to participate, and exclusion criteria are for mean that you cannot participate. So this was asked at the previous session. You have to be an adult between 18 and 65 years old. Obviously, this is a therapy that one would hope to study in children, but as in terms of how you conduct clinical trials in general, the FDA and the regulatory authorities want it to be trialed in adults first before it's tried in children. You have to have a clinical diagnosis of PCD, and I know there's a lot of information on the PCD Foundation website about these potential confirmatory tests and how they're conducted. Basically, they help measure the function of your cilia and using nasal nitric oxide measurements, or actually looking at pictures of brushings of your cilia to see how they do or do not beat properly. And you do have to have PCD caused by the select mutations in the DNAI1. That's because this messenger RNA is specific for that particular defect. I know that everyone is anxious that therapies for individual variants amongst patients with PCD be developed, and I know that that's in the works, but you have to start with one thing at a time, and this is that first study. Lung function has to be not too bad, basically, and generally pretty good. We don't want to put people in the study who already have pretty bad lung function, for whom this could be unsafe or just not. It certainly wouldn't be effective in one dose. And like most studies, pregnancy and contraception, we have to avoid pregnancy during a trial. Basically, exclusion, if you're very sick, if you have a very other serious medical condition that's untreated and that might put you at risk, certainly we don't want you to be included. If you have an active infection, there are some specific things. If you've had a vaccine within 2 weeks, you just need to wait a little longer to do it. Smoking, excuse me, is a disqualification, and an inability or unwillingness to collaborate and do the actual parts of the study. Obviously, it wouldn't be for you. So this will also, again, this is information that will be provided by the company in great detail to anyone interested. Next slide. So how many patients are needed? We don't need many, but it's so important to get moving, because this process will take the company closer to getting a drug that's available to patients. So in the first cohort, 3 patients with DNAI1 are needed. And then in the second cohort, 3 to 6 participants with the same genetic mutation. So not a lot of people, but time is of the essence to move this into the clinic where more patients might have access to trials. So that's what ReCode is looking for. And I know they have a lot of people devoted to trying to move this along as quickly as possible. So next slide. A little bit about the timeline of the trial. And this is really from the perspective of what might it involve for you. And I mentioned it in words, but it's nice to see it over a timeline. So if you are interested and you would like to be considered, and you haven't, wouldn't obviously be excluded for any of the reasons that I discussed, you would have a screening visit. And there's a window of time. If you know your mutation status, there's a 26-day screening window. If you don't know, there's a 3-month screening window, and all of that could actually be obtained if that's something that you would like to do. So the treatment, you have a visit the day before the treatment, and then on you get the next day, you get just one inhalation of the drug. You stay overnight, and then you're able to leave. Follow-up can be done at the same unit, or on day 3 it needs to be done in that unit. But thereafter, you can do the visits remotely if it's not possible for you to travel back to the study site. So there are visits on day 8, 15, 29, and then there's quite a gap, 3 months, and then a 6-month visit. And across all of this, all the information will be collected to monitor you for any concerns or any questions that come up. So that's the timeline. So I think the burden on patients in terms of studies that I've seen, 1 day in person, a day and a night, is pretty good. And it really is just one inhalation. And there will be some testing done. I think that's on the next slide, or maybe not. Yeah, let's see. Next slide. Yeah, so this is what's going to happen at the visits. At every visit, you'll receive an exam and some vital signs. I believe that you will have, not on all the visits, but on many of the visits, spirometry and lung function tests, which many of you may already be familiar with. An EKG probably at the outset, just to be sure that, there's no problem there, and a chest X-ray to be sure you don't have any intercurrent infection or issues that might interfere with your participation. There will be some blood tests and urine tests. Again, not every single visit, every single test, but that would probably be the most inconvenient thing for you, is the blood and urine. And the nasal brushing, which is just done, I believe, at the first study, and John's nodding. Now, I just want to say a word for those of us who've done COVID tests. This is nothing like the COVID stick it as far back in your nose as you can test. This is really just the outside of the nose. So don't have any concerns about that. I felt sometimes with the COVID test that it was not very comfortable, but this is not that. It's just on the outside of the on the inside of the nose, just at the front. So next slide. So what happens after the study ends? Well, the company takes, as highly orchestrated and dictated by the FDA, they look at all the data, they collate it, and then we'll likely share it through publications and professional conferences. None of your personal information will be shared. It's de-identified data. You become a number and it's separated so that there's no way that anybody would know that you had participated or whose data belonged to whom. The data then, once it's actually in the process, it may be shared in public, but meanwhile, the data has to be shared with the FDA and to regulatory authorities, potentially in other parts of the world, as part of the package to keep moving, we hope, this trial forward closer to patients. There is a possibility for patients who are in this trial to participate in future phases of the program, for instance, when repeat doses are given, and future clinical trials will be forthcoming. Next slide. So this is probably the slide that everybody really wants to see. Exciting news is that thanks to ReCode working very fast, they are on the verge of opening the site for the study. I'm told that it is in Las Vegas, and which is easy to get to for those of you who might have to fly in, which I assume could be some folks. The government requires that all clinical trials are kept on a site called clinicaltrials.gov. I don't know if it's posted there yet, but it will be posted. But probably your best source, if you have questions or where can I find out more, is the email, clinicaltrials@recodetx.com, because they are, in fact, pushing all of this out to these sites and working every day on making sure everything's ready to go. You could maybe contacted to be pre-screened. You can do that over the phone and find out if this is something for you. If you are contacted, you will get more, like I said, more information about the study. And then as you hear more about it, you can choose to sign the informed consent to participate or not, depending on how it sits with you. So again, I think there'll be a lot of it. This is very much on the cutting edge. This is just coming out that this trial has gotten the IND and the go-ahead. And I know that things are in motion. And I know that ReCode is hoping that perhaps we can at least get a few patients in before the end of the year. So I'm wishing them luck on that. And I think it's really in the hands of the patient community at this time. Your participation is critical at this point to how things will move forward. So next slide, and I think we're going to open it up for questions. Yeah, great. So I hope that was helpful. I'm happy to take questions, and Dr. Matthews, who represents the company, is also, and as well as Michelle, who know, and Lynn, who know a ton at this point. They are experts as well in PCD and clinical trials.
Yeah, please put your questions in the Q&A thing, and we will try to field them and get them to the appropriate person. We have a question. Just saw the FDA approve the first medicine using CRISPR technology. Would be curious if this is at all positive for mRNA therapies.
Yeah, I just, funny, I think that was, people were so excited. So CRISPR technology, I think it's exciting because it just is taken so long, and now we're actually having a treatment, right? So the CRISPR technology is approved, and what CRISPR does is actually go into the DNA, and it actually engineers and cuts out and then puts in things that are missing from DNA. And I would just say that in the sense of positive for mRNA, I mean, like I said, mRNA doesn't do that, but I think, unfortunately, at the time of the pandemic, there was a lot of misinformation, or I think people were concerned, mRNA, what does that mean? I think as science moves forward and as people have a growing understanding of the potential of these medicines and more security around it, and we stick to the facts, I think that it just bodes well for the hope of these treatments is to really ultimately cure patients and get at the root cause. So I think it's exciting. I've treated patients with sickle cell. This is a chance for these patients to live a normal life. So I think it doesn't have direct implications because this is a different type of technology, but I would just say in the field of genetic medicine that it's exciting. Gives us hope.
I think this may have been answered in your last slide, but will participation exclude future criteria?
Absolutely not. So that's great. Actually, I think we're going to count on it because this is a rare disease.
And then we have a question from a patient community. So we have some from physicians here, and I'll try to let you know who is who. The last question was from a physician. What does ReCode expect to learn from N equal 3 to 6 that cannot be learned from safety testing in the general non-PCD population? Yeah,
that's a great question, and John can take it. But what I'll say is this: is that you have to see a signal. I mean, this drug has been designed to do this thing, right? Healthy volunteers have have a normal function. So you really want to see, if we give this, do we potentially see the thing change, right? Do we measure that? But more importantly, I mean, patients are patients. These patients may have underlying lung disease, and you learn a lot from safety testing already, and there has already been a healthy volunteer study. But I think that it's important to do it in patients who actually have the defect. John, I don't know if you wanted to answer any more fulsomely or not.
No, I would just, excuse me, just add, I mean, this is such a specific treatment. The mRNA is the full wild type instruction for DNAI1. And so to give it to people who don't have that problem in PCD wouldn't be appropriate. So we're directing it very much to get the safety experience via the nebulized route. So it's inhaled into the lung. That's specific for DNAI1, and allow us to gain that experience. I mean, there is a potential risk that you could have drug antibodies. We think it'll be unlikely that they would be clinically significant, but we have to measure them, and so it becomes very relevant to test it in the patient group that are going to benefit most. The benefit risk equation that we talk about. When we give a drug, we have to expect to have benefit over any potential risks.
Yeah, great. No, good question. We got some more.
Lots of good questions again.
Great.
So we have a question. You've mentioned there are over 50 causal genes. What stops you running a trial that looks at several of these in one go rather than just DNAI1? Do you foresee expanding into other genes in the near future, or does it rely on the outcome of the DNAI1 trial?
Well, A lot relies on that, not only the results, but also if we can continue to attract, I'm saying the company can continue, as a drug developer, can continue to attract investment. Listen, everybody would love to just have the treatment for all of the genetic variations, but you have to start somewhere. And if you have a different variation, you have to design and test in a preclinical model, the DNAI1 or the DNAH5, whichever variant. And this is the one that's coming through first. I know that it is the hope that the other genes will have similar targeted mRNA treatments. So yes, I would love to just, if this therapy was felt to have any chance, but it has to be specific for your version, very much personalized medicine.
We have several questions from some of our clinical centers directors who are on this webinar.
Great.
The first one, does the patient need to have biallelic pathogenic mutations in DNAI1?
Yes.
Okay.
The second one, will the company pay for travel and the stays in Nevada? Any hope for a second research site on the East Coast?
The first answer is yes, the company will pay for all the expenses involved. I don't know about the plan for a second research site on the East Coast. I don't think there is one right now, but
yeah, we're starting off at a phase 1 unit in Las Vegas, and that's partly because the speed at which you can get a central ethics committee to get a site up and running. We are collaborating to get an additional center up and running in the US.
right.
I've just realized I should add that the previous question, biallelic pathogenic mutations of DNAI1, they do have to be pathogenic mutations, but the definition biallelic has a very scientific meaning in terms of needing to know the parental genetics. I think what we say in the protocol is that the principal investigator has to have evidence of a pathogenic mutation in DNAI1 that's of pathogenic significance. So that would mean that there would be mutations on both alleles.
Okay.
This is from a center director who has done a lot of nasal scraping. He says, does the nasal brushing require collection of nasal epithelial cells for cell culture? That will probably require more than just a simple swipe of the anterior part of the nose.
Yeah, so this is at screening. It's enabled us to get some primary tissue. So you're right. It would be a brushing equivalent to the brushing that's done for diagnostic purposes. The cells and the brushing we put into some media to allow organoids to be developed.
I probably got you. I got you into this trouble, John, because all I know is it's not as bad as
we weren't pushing a probe into the deep part of the nose.
Apologies, there is someone who hasn't done the nasal brushings, but I just know that it's I have a little PTSD from the COVID. Anyway, let's carry on. These are great questions, and John, I'm glad you're on the call because you've actually done these and I haven't.
In the early years at our family education days, everybody got scraped all the time, and we would walk around with our noses covered just to keep probes away. So,
okay, so this is an audience that appreciates that.
Okay, another question: what are the key things you hope to learn from a single dose trial?
Key thing is safety.
Yeah,
I mean, I think that's really front and center is that the safety experience will allow us to go forward and to do repeat dosing. But in all drug development, we start with a single dose and get that safety experience.
And I'll just add, as a pulmonologist, whenever you nebulize things and put them in the lungs, which have their own immune system, yeah, I'm always concerned that people don't have any type of adverse reaction. So I think that's prudent and not uncommon to do this in this way for an inhaled therapy.
I think we've answered the question about is travel to Las Vegas for now, and ReCode covers the expenses. Another question: is there preclinical data that you could share in terms of safety?
I think there's preclinical and there's clinical data from a healthy volunteer study. John, I don't know where that is. So it has actually been The
Yeah, I would just
well,
say, so that this study in the US, we've submitted it as an IND to the FDA. And so our single dose toxicology has been reviewed by the FDA in 2 species. And we have completed, it's an ongoing study. It's still technically blinded, technically because it is blinded. And so it's an ongoing part A to this single dose healthy volunteer part of the protocol, part A. That's up on clinicaltrials.gov. And as soon as we can share the data, we can. For the purposes of participants, they will see in the informed consent the summary of safety, and they can ask questions and be provided with more information from the investigator brochure that has a summary of all of the safety experience in the healthies.
Yeah, I am right. It's more detailed, but I had some acquaintance with some of the summary, and there's you know, I think there was not anything there to be concerned about, but more details to come, obviously.
We have
Great.
a question if this trial would be possibly considered for FDA's accelerated approval program.
Wow. John, you want to comment?
That's yeah, it's kind of regulatory question. We've not submitted for fast track designation or breakthrough status. I think those things you need to have some evidence before you can have a package to submit. So this specific trial at the moment isn't under any of those programs.
Is there a chance for future therapy for the LRRC6 gene?
Well, I mean, John can answer, but I mean, I think if in principle this approach works, I think it would certainly be the goal to pursue the other gene mutations with similar correction. John, I don't know if you want to comment. I don't know anything about that mutation specifically, but again, this has to make it through the path, and then the plan is absolutely, I think, to investigate treatments for the other mutations.
Yeah, I don't actually know that specific gene mutation. You know, on our website we describe genes 1, 2, and 3. So the plan is if the messenger RNA for DNAI1 is successful and we can ungate additional genes, then yes, the aim would be to treat as many as people as would be possible.
Yeah. Does the mRNA therapy only impact the lung since it's nebulized, or will other cilia be affected?
That's a good question, and John, you can jump in. But basically,
impacting the lung, yeah,
it'll impact. Yeah, it's just going to impact the lung. There's no systemic effect. I think for treatments in those areas, you would have to target those organs and those sites in the body.
Is it realistic to think that cilia that have not been beating normally for decades would be able to function normally with correct coding, thinking atrophy, etc.? Maybe this gets to the fact that cilia generate and. Yeah.
Well, you know, that's an interesting question. I and John will have a different take on it, I'm sure, Dr. Matthews, than I do. But I think of it as like you've got this complex machinery with a spanner in the works, a protein that doesn't work. And you're correcting that. Now, the state of the rest of the cilia, I don't have a lot of information that there's such degeneration in the rest of the mechanics that fixing this one piece wouldn't work. And as a matter of fact, there is preclinical data. I don't know that the animals and the preps were decades old, but there is good, solid preclinical data showing correction from not beating normally to beating normally, to suggest that this would work. I actually don't know if cilia atrophy, John, but I don't
think they do. I don't
think they really do. I think
turnover of cells in the airway.
Yeah,
you know, when we get viral infections, right? we can all lose cilia, epithelial cells shed during a viral infection.
That's right.
And so I think the body does have amazing regenerative capacity in their basal cells that are the progenitor cells for ciliated cells. Right. And so the idea would be that the message remarkably is brought up into the axoneme as it's developed in new
As it's growing.
ciliated epithelial
Good.
cells. I don't
Right.
think that's going to be a problem.
Yeah. I mean, to that, the turnover of cells, I mean, red blood cells last 120 days. We're always making new things. So
Yeah, there aren't cilia that have been there for decades atrophying.
yes, that's yeah. Thank you, Michelle. That's the obvious answer. Yeah.
Will the trial be limited to only 3 to 6 participants, or would more be included if there are more people interested?
That would be a great problem to have, John.
Yeah, I think part of the samples, the number of 3 in each cohort and expansion to 6 in the second is partly because it's a rare disease with a number of available DNAI1 patients, relatively with only 10% of a rare disease. We're dealing with small numbers. I think the expansion will come when we start to do repeat dose studies and start to measure benefits on lung function and improvements in mucociliary clearance, where we'll start to need protocols with 10 and 20 subjects, and phase 2 studies will probably have to be larger, of the order of 20 subjects per arm, so 40 to patient studies. So there will be definitely opportunities for more participation, but until you've completed your single dose, you can't proceed.
We have a question from a patient. All 3 of my grown children have PCD. For one, the cilia move randomly. For the other 2, the cilia don't move at all. Could they all have the same, and then she corrects at the bottom, the same DNAI1 gene at play here, causing all the problems?
John, Dr. Matthews.
I would say genetic testing is becoming more of a feature that we'd like to see for all patients with PCD, such that we could get to the very precise genetic cause of every individual. And so DNAI1 generally have the most severe cilia beat reduction, such that they generally don't move at all, and occasionally there's a flicker in like 1 in 100 cilia. So I would say, given the description there of your grown-up children, I would have an open mind as to what the genetic diagnosis is until you do genetic testing. We've set up a website called Think PCD, and so we are offering the opportunity to get genetic testing for people with PCD. So I would encourage, and we'll hopefully share more information about Think PCD with another one of these webinars in the new year. So we'll come back, but I would have an open mind and do try and get a genetic diagnosis, as it will only encourage us, as we have registries, we have more and more granularity to the genetic causes of PCD, the more ability we will be to develop specific treatments.
I would say, I don't think, and Mickey Lee is on, she can correct me if I'm wrong here, I don't think we've seen a family where there's multiple affected children and the children have PCD by different genetic causes. If all 3 of them are affected, it's almost certainly the same genetic cause, until proven otherwise. But we will also share all of this information about Think PCD and how you get in touch with ReCode. We'll make sure that's available to the patient community. And just want to reinforce what Dr. Matthews said, what Dr. Doyle said. All of these therapies are going to rely on knowing your genes. So gene testing is going to be critically important for our population going forward, even if not for this study, just in general, gene-based therapies are the way things are going. So consider taking advantage of getting genetic testing. We have a question, will inclusion criteria always require minimum 50% volume?
And I can take that. I would imagine, John, that's just to keep the sickest patients out of the study, right? At some point, you might consider the inclusion being different.
Yeah, I think the aim would be to expand to be able to have safety in the entire PCD DNAI1 population. So yes, we would expand that as we get more experience.
Yeah, and just a reminder that the point of this first small study is to establish safety. So the inclusion criteria are made with that in mind, and the follow-on studies may be a little bit different. So don't think you're completely excluded for the future just because you don't meet inclusion criteria now.
Right now, that's a great point. Yeah.
Another question about DNAI1. When might we see other studies for genes like DNAH5?
Well, John will answer this on behalf of ReCode, but I'll answer it in terms of a drug developer. I think that this all depends on how quickly we can get the first proof of concept through, and then get others incentivized to really start moving for the other genetics. I don't know what ReCode has in its back pocket, but I could say this could open the floodgates for the different therapies to be addressed. And John, you may have some more specific things to say, but I'm hopeful it'll be sooner rather than later now. We just have to get the first one done.
DNAH5 accounts for 20% of PCD. And I think there's no technical reason why we couldn't have a mRNA for DNAH5. So I don't want to overpromise, and certainly it won't. There are no clinical trials planned for next year, but I hope that with engagement and DNAI1 success, that we'll be able to march through certainly the most frequent mRNA gene loss of function genes.
We have a question about DNAH11. I think it'd be helpful here. The question is asking if there are 50 variants, and I think we need to explain the difference between a gene and a variant. So she said, if there are 50 variants, will there be trials for each? It seems like that would take a long time.
Yeah, so for DNAI1, there are 130 pathogenic mutations. So there are multiple variants that are these mutations that can lead to loss of function of the protein for the DNAI1 gene. So the mRNA treatments are, we call it agnostic. It doesn't matter which mutation you have. We don't have to do trials mutation by mutation. It's loss of gene function by gene. So we can replace the full gene for DNAI1. So you would go gene by gene to be able to provide effectively a replacement or a correction by replacement with the mRNA.
I just want to say, this is one of the things that's exciting about this to me. So DNAH5, we don't know how many of these are actually variants or mutations that cause disease, but there's been over 4,000 variants on that one gene. So if we had to be doing this variant by variant instead of by a whole gene, it would really be challenging. So this is gene based and not variant based. Again, we're having more questions about specific genes, which I think we've probably already addressed. Will inclusion be limited to nonsense or frame shift, e.g. testing immune response for introduction of DNAI1?
No, so as long as it's a pathogenic mutation, which would include nonsense or frame shift mutations, I think the question is gated around, could there be some small amount of DNAI1 that might have antibodies against it? I think that's part of the experience that we need to gain with the safety, but there's no specific exclusions for nonsense or any specific frameshift mutations.
Sorry if you mentioned this. Is the trial open to non-US citizens?
The trial is being conducted in the US, in Las Vegas, at a specific site under the IND. So to that extent, it's for people in the US. I don't think there are any specific regulations about having to be a US citizen to participate in the trial.
I think, yeah. I don't think there's...
I'm sorry.
No, I don't think there's any limitation there, John. It's conducted in the US, and they have to go by the laws and rules of the US if they're a participant in a US study. So they should be able to participate.
I would say, given the duration of the trial of 6 months, you would have to have the ability to reside in the US for the duration of the trial. I mean,
would they not be able to come and go? Would they not be able to come and go? Something to think about. Anyway,
yeah.
It would be a good problem to have if we have that many patients interested.
But I do think it brings up a good point, just so patients understand that these regulatory bodies, what we're talking about here is an FDA plan for getting to approval, and every country has their own regulatory authorities, and they're often very similar, but they're not identical. So what we're talking about here is a US trial.
And we do have a site in the UK that will be opening in the new year, and also in Sydney, Australia.
No, I love this question. What can the families of the patient population actively do to facilitate the forward momentum of clinical trials?
Wow. Well, you're doing it. First of all, you're involved with a great patient advocacy organization who should remain your trusted conduit for information about trials. I think, we don't know if people have it or not in the general population. Why did we not do not diagnose more patients? Because we didn't have anything to offer them except symptomatic treatment, right? We started testing for CF when we had CF specific drugs. And so we do it routinely. I think PCD has to set out on that journey. So I think genetic testing, promoting education and awareness, promoting participation in clinical trials. Those are my thoughts, but I'm sure Michelle and Dr. Matthews have some other thoughts. But it's just great. I mean, you're already doing it because you're involved in leaning in.
I think peer-to-peer is so important, and getting involved with us, with the PCD Foundation. The message needs to be that this is on us. Nobody else can do clinical trials for PCD except for our population. So
right.
The more we can be united in that messaging with each other and encourage each other to be more involved, I think the better. We have some opportunities here that we could only have dreamed about 20 years ago, and we want to make sure we don't miss these opportunities. Okay. Is there an idea how often the therapy would have to be inhaled?
Since the mRNA treatment that is not permanent, if approved, the drug will require multiple doses. So we do know it'll have to be taken multiple times. We do not know yet the dosing frequency or the schedule. This will be determined through our clinical trials.
That's something that typically gets explored in the next phases. You'd start with the single dose and then you experiment around. Obviously, you want to give it where it works the most, but not more than you have to and what's convenient for patients. I can't imagine, and John, I'm speculating because I don't know. I'm sure it's not more than once a day, and it's probably not even daily, just given the what I understand of the biology. But that's all to be determined, as John said, as Dr. Matthews said, in the trials upcoming.
I think this probably is a question that would be better for Dr. Lee, a breakdown of mutations and their percentages. So we're talking again about genes and not mutations. And we do have a breakdown of how common different genes are. The questioner says, I'm worried that my genetic mutation may be so rare that they are part of the long tail that was referenced in the last session. And some of these more rare PCD mutation or genes may be not in the first round of development for therapies. Would that be a fair statement?
Well, I mean, I'll take a swing at that, and then maybe Dr. Matthews can answer. I think that this all depends on how quickly we can get through the trials and show proof of concept. And frankly, I mean, ReCode's a startup. If they can show, and if they could have a lot more resources and money to apply to this, to start this pipeline moving faster. I don't know what the time frame. I don't know how long that tail will be. It'll depend somewhat on the results that are going on. But I know that no one would like to go faster and cover more ground more quickly than the company currently. It's just a question of a lot of the information that you get from the trials and how the time frames go. So I would not be worried or lose hope. I would be very hopeful that this is now, it's happening right now. It's starting to happen, and it's still to be determined the time frame for bringing other gene corrections to PCD patients. I don't know, John, if you want to add anything.
Yeah, I mean, I think it's important not to overpromise in like the near term, because there are there's scientific issues around knowing exactly how to help a specific condition. And that takes a lot of time and research. So I don't want to overpromise. However, I will say in my general reading of rare disease, I'm constantly pleasantly surprised that there are huge efforts now to show that if there's a willingness to even treat an individual patient, then there are some rare advocacy groups that have formed around treating N of one, a single individual.
That's right.
And so I've learned not to make any assumptions. I think science takes the lead, and I think patient advocacy will drive science. And I think but I'd be fair to not overpromise that it's not going to happen in the next couple of years for these mutations that are loss of function genes that are the less frequent ones. I think that would be fair to say.
I'm cognizant of our time here. We've got about 5 minutes left and a lot of questions still to answer. There is one that I want to address very specifically. How is the trial being funded? Is it ReCode or the PCD Foundation? It is not the PCD Foundation. We would not even have the bandwidth to sponsor a trial at this time financially. So that gets back to sponsorship. The sponsor for this trial is ReCode.
ReCode.
Do we have a summary of trials that have successfully used mRNA therapy for other health conditions? And this is the same questioner. Would the genetic testing be available in the UK as well, or and test sites in the UK, or would people have to travel to the US for participation?
John, you can take the second. Well, you can take that question, I think.
I think if you're in the UK, I think that the different mechanisms for getting genetic testing, and certainly the intention wouldn't be that you'd have to travel to participate. So yeah, if you're UK based, then the genetic testing can be provided through the NHS, and so there is an ability to participate in the UK.
And what was the first? I'm sorry, go ahead.
I just wanted to mention, we have just heard from our partners in the UK that PCD is going to be on the newborn screening panels in the UK, 36G.
So awesome.
So that is amazing.
Amazing. Great news.
Would participants need to stop their normal medication for the duration of the study period?
No, that's not the intent. The aim will be to keep existing treatments going.
Are there any known side effects at this time?
So we have the experience in the healthy volunteers, and those will be described in the informed consent form. I think it's important that the participants see what's in the informed consent form and can ask questions with the principal investigator at the site to better understand any adverse events. The adverse events in the healthy volunteers were all mild in severity, and the study's still blinded, so there's limited description I can give to them at the moment. There was one moderate, somebody had a COVID infection at day 15, which was not
unrelated.
An unrelated adverse event.
The second part of that other question, I just wanted to get back. Somebody said, are there other mRNA therapies to compare to? Yes, there are mRNA therapies. Probably the most widely known one are the COVID vaccines. But I don't know, John, if you have any other information on mRNA. I know there are a lot of people in the space developing therapies, but do you guys have a particular list of other ones that are already available? There's one
approved mRNA by the FDA.
Yeah, I can't remember it right off the bat, but I would say that the COVID vaccine experience, I mean, it's worldwide. It's been given to hundreds of thousands of people, millions of people. But there's more coming. There's a lot of this in process, under study.
In terms of the percentages of people with each gene, is that worldwide or is that just US percentages? 10% have DNAI1, 20% have DNAI1.
I think it's probably worldwide, isn't it, John? Or it does vary by country, but it's so rare. You have to collate all the epidemiology from across the world. I don't think it's been broken down by country, has it?
There's a lot of research to try and get a better handle on what the exact percentage is. So I think 10% is a general number that could apply to both the US and worldwide at the moment, but that may change.
I mean, I think for the patient community, we need to understand that we have only identified a small fraction of patients with PCD. So whatever we have for statistics and demographics right now, that is as
Inadequate.
now, and it can evolve and change over time.
Yeah.
I think we're getting the signal that our time is up. So thank you all so much for joining us today, and for our speakers and presenters. Thanks very much. We really appreciate you taking the time to talk to the patient population and tell us about this exciting opportunity. We will share information on our foundation, YouTube and social media pages, and send information to you regarding some of the links that were in our presentations today.
Happy holidays, everybody, and good luck. Thank you all so much. Take good care.
Thanks. Bye-bye.